Hello, Curious Molecules. Welcome back to the channel. If you're new to this channel, you are also very welcome to Curious Molecules. If you're here, you're a curious molecule. You've surely wondered once or twice or as many times as you can't count in your life what really happens in my body when I eat. Where does this food go to? What happens? How is it broken down? So you're in the right place where we make biochemistry and all the complex molecules and reactions very, very simple for you to understand. As simple as eating your bowl of rice, which is ironically what we are going to be talking about today. Definitely not rice, but what happens when you eat that rice. So today we are going to be talking on digestion of carbohydrates. So if you haven't watched the previous videos, you should, because we gave a brief introduction into what biochemistry is, how it applies to your daily life, how you use it, and how it's going to help you to understand other things better at the molecular level. So you should definitely watch that video. There's also a video on the introduction into carbohydrates. So if you haven't watched, you should watch so that you can follow up from there. So we have seen or heard probably before that carbohydrate digestion or digestion generally starts in the mouth. But today we are going to see the stages of digestion of carbohydrates in the body. So there are three phases: the salivary phase, the pancreatic, and the intestinal phase. We're going to see that all these stages occur in two parts of the digestive system. The first part is the mouth. In the mouth, we know what happens in the mouth, the chewing. So the mouth is like the blender. The teeth help to blend those large chunks into something smaller, not necessarily as small as possible. That is what happens physically. But now we are talking biochemistry, remember, at the molecular level. So, what happens to those molecules? You also have blenders. In this case, the molecular blenders are enzymes. They are going to break down those large molecules into smaller ones. If you watch in the video describing carbohydrates, you'll see that there are small single unit carbohydrates called monosaccharides. That is what the body can absorb and use to produce energy and other processes. But from the food we eat, we have larger units like polysaccharides, starch, that we saw in the previous videos. So the mouth has to involve something that can break down these large units. The salivary glands produces an enzyme called salivary amylase, and it's going to break down those large units. So those large units can reach thousands, hundreds of thousands of units long. So from the mouth, the food moves down to the stomach. But in the stomach, there is no digestion of carbohydrates there. So in the comment section, I want you to comment the reason why there is no digestion of carbohydrates in the stomach. All right. From the stomach, we move to the small intestines. Digestion is said to continue in the small intestines. So here you can be wondering, we are saying pancreatic phase. The pancreas provide the environment and the materials, needed for digestion. So the pancreas are going to provide amylase again, but in this case, the amylase is pancreatic amylase, not salivary. So it's going to perform the same function, but in this case, it's breaking it down into shorter chains, breaking it down into single units for some. So just imagine in your blender, if you've put in some beans or whatever you want to blend, at a certain point in time, if you remove that whatever food substance you have there, there are going to be some that have been blended to smooth, others will just be chunks, others will be half, others will still be full. So that's what happens during digestion. The enzyme keeps doing it over and over till all the food substance has become single units. But sometimes they end up being disaccharides or two sugar units. And disaccharides can be broken down into their corresponding monosaccharide units by specific enzymes. You have lactase for lactose, maltase for maltose, and sucrase for sucrose. At the end of the day, you have just single units of carbohydrates in the small intestines for the body to absorb. After the body has absorbed what it needed, of course, the rest of it is to be thrown out as feces. So there is no digestion that occurs there. And here, the liver is mentioned here, the pancreas is mentioned here. I want you in the comment section to tell me how the liver aids or how does it contribute in these digestive phases that we are talking about here. All right. After digesting all of that, we said at the end we want them to be small single units so that they can pass through the intestines into the bloodstream. So the movement from the small intestine to the bloodstream is what we call absorption. So for carbohydrates, they are all monosaccharides, definitely. So they are initially absorbed by a common transporter. So the small intestine here is acting like an international gate in the airport. So it's going to allow just some particular, each gate has maybe the particular passenger that can pass. In this case, there's a common gate. Let's say everybody passes from that gate, but they have to go to uh, the same destination, which is the bloodstream. They're going to different parts of the country. So here, the GLUT5 is the common transporter, which we see here. Glucose, fructose, galactose can pass through. But also there is a transporter which is specific to glucose and galactose, which is the SGLT transporter. This S is represents sodium. So this particular transporter or this particular gate requires sodium for them to pass. So if there is no sodium in the body, these are not going to pass, and so the person will have a poor absorption of glucose and galactose. It's true they are going to be able to pass here through, but there's a reason why the body has those transporters to be able to meet the needs of the body. Okay, so at the end of the day, you have all of those monosaccharides have moved from the intestines into the bloodstream. That is what we call the absorption. But there are some carbohydrates that cannot be digested in the first place, not to talk of absorbing them. Something like cellulose cannot be digested by humans. But there is a particular animal that can digest cellulose, and I want you to comment that animal in the comment section. We are coming to the interesting part, metabolism of carbohydrates. So after you have eaten, digested, taken in what is necessary through the intestines into the bloodstream, and excreted what you don't need, what happens? So metabolism happens to all molecules. But for carbohydrates, it is the breakdown and build up reactions that involve carbohydrates in the body. In the body, you're going to hear of several metabolic pathways like glycolysis, which is very old metabolic pathway, old but useful, because we are going to see that in the next slides how useful glycolysis is. We also have gluconeogenesis, glycogenesis, glycogenolysis, tricarboxylic acid cycle, and all those pathways. So in the comment section, as usual, you should comment your most preferred pathway for any reason. It could be because it's simple, it could be because you feel it's very important. You could have a single molecule in that pathway that you love so much, and any reason, comment. So now to one of the most important pathways of metabolism for carbohydrates, glycolysis. Glycolysis, also known as the Emden-Meyerhof pathway, after the people who discovered this pathway. So what is glycolysis? It is a major pathway for glucose metabolism. It's a pathway where glucose is converted to pyruvate or lactate, or is converted to pyruvate basically, and then the pyruvate is converted to lactate depending on the oxygen status of the body. So why is this particular pathway important? So glycolysis, as the one of the first pathways, metabolic pathways that was discovered, has been seen as the principal route, the major route through which glucose, fructose, galactose, and other carbohydrates are metabolized or are used. So we are going to see in the pathway that glucose, fructose, and galactose can or are involved in this pathway. And we said that at the end of digestion, you have these three. So if you have them, what are you going to do with them? You should be able to use them. If not, they accumulate in the blood and they cause problems. So glycolysis is a major route for consumption of these carbohydrates. It is also very important because it operates in aerobic and anaerobic conditions. There are some pathways in the body that if there is no oxygen, they are not going to occur. But glycolysis is not one of those. So whether there is oxygen, whether there is no oxygen, glycolysis would occur. The only problem is maybe the byproducts at the end of the day. And also, in the absence of oxygen, lactate, which is produced as a byproduct, can be used to produce glucose in case of starvation or lack of glucose in the body. The liver undergoes gluconeogenesis, and lactate is a precursor for producing glucose through gluconeogenesis. And we said it can occur in both oxygen and non-oxygen conditions. And we can take, for example, skeletal muscles in the body during strenuous exercise. If you're running or you're doing exercise that is stressing the muscles a lot, or if you're lifting weights, all of that, anything that stresses the muscle the muscle is going to be breaking down a lot of glucose to produce energy for you. But also, in that excess or strenuous exercise, there's also limited oxygen going to your muscle. That's why you would have cramps, for example, because there's limited oxygen, but there is also energy still needed. And if the body couldn't perform anaerobic glycolysis, you wouldn't have energy during exercise, that's strenuous exercise, because there's limited oxygen. So this is very important for skeletal muscles. Also, glycolysis has enzymes which, like hexokinase, which when inhibited are going to favor storage of glucose in the form of glycogen. This goes to say that glycolytic enzymes have very important roles in other pathways like glycogenesis. And deficiency of some of these enzymes could result in hemolytic anemia because we know that the red blood cells can get their energy only from glycolysis. Why is that? Answers in the comment section. Why can red blood cells get energy only from glycolysis, not other pathways? Right. So we are going to the process of glycolysis itself, the step by step process of glycolysis. So, glycolysis, as we said, is a pathway where glucose is broken down to pyruvate. So, we see here that glucose is broken down to two pyruvate molecules. And we also see some highlighted molecules or intermediates, as we can say. The energy currency of the body is ATP. So, ATP here is used and also produced. So, we could divide or we could classify the whole process of glycolysis into two phases. One is the energy investment phase, which is this one, energy, ATP. Investment, if you're investing, meaning you're putting in something to gain more at the end. So you have investment phase and the yielding phase, energy yielding phase, this phase. So at the beginning of glycolysis, two ATP molecules are invested, and at the end you have four ATP molecules. So at the end of the day, there is a net benefit of energy from this pathway. All right, the pathway, as you can see, has 10 steps. We are not going to go deep into the 10 steps. If you want, for more understanding, I could do another video just on the steps of glycolysis. But here I just wanted to highlight the major molecules in this pathway, which is the energy, which is what the pathway is aimed at producing. So as we said, this pathway is a very important pathway, which means that most cells or almost all cells in the body undergo glycolysis, and it occurs in the cytoplasm of cells. Generally, we have examples or we have important enzymes that are involved in the pathway. If you notice, every pathway or every step, in the pathway that involves ATP, the name of the enzyme has kinase, hexokinase, glucokinase, fructokinase 1, phosphoglycerate kinase, and pyruvate kinase. So kinases are involved, or enzymes that are involved in steps or reactions that involve ATP production or usage. That's something you can take note of. Usually, when you want to measure blood glucose from a sample, there is a particular tube, a fluoride tube, that is used to collect that sample. The major aim of collecting in that fluoride tube is to prevent breakdown of the glucose. You want to measure glucose, so you don't want it to be breaking down. So that fluoride usually binds to this enzyme to prevent breakdown of glucose. The next thing we have to note about this cycle is the rate determining reaction. A rate determining reaction is a reaction that determines how fast or how slow that reaction should occur. So if you are in dire need of energy, like during exercise, your muscle cells need a lot of energy, which those enzymes are going to increase the rate of synthesis. So they're going to make this glycolysis move faster to produce the energy that you need. If you're resting, for example, you don't need a lot of energy at that time. These same enzymes are going to tell the glycolysis pathway to slow down. So those are rate determining reactions. We have three of them: hexokinase, we have the phosphofructokinase 1, and we have pyruvate kinase. So if the body wants to regulate glycolysis, they usually affect these enzymes by telling them to slow down synthesis or increase synthesis depending on the needs of the body. All right. So we're just talking about regulation. which occurs in the cycle by enzymes like this hexokinase, phosphofructokinase 1, and pyruvate kinase. Now we saw this hexokinase and glucokinase in the pathway here. We are seeing that the two synthesize the same path, the same reaction. So I want you to let me know what the difference is between this and this. If they synthesize the same reaction, why do they exist as two? Why not just one of them function? So what is the difference? If you want to regulate a pathway, you have to have a signal. So if I want you to stop running, if your body wants you to stop running, it's going to give you a signal that you should stop running by getting weak, and that's physical. At the molecular level, there are signals that are going to help determine the rate of the reaction. So the enzymes are there. The enzymes are there to directly impact the pathway. But for the enzymes to say, okay, I'm going to reduce my rate of synthesis, they have to be triggered by a signal. So for example, hexokinase, we have seen that it's in the first reaction in the glycolytic pathway. So it's encouraging glycolysis to go on. Activation, the signal for activation will be definitely done by insulin and these other molecules we're going to see. Let's take insulin, for example. Insulin is produced when there is excess blood glucose. If you've just eaten your bowl of rice, again, our jollof rice, if you've just eaten your bowl of rice, your body has excess glucose in the blood after about 1 to 2 hours. Within 1 to 2 hours, sorry, your blood glucose rises. And when blood glucose rises, insulin is produced. Insulin goes to the pathway and tells hexokinase, you see, there's a lot of glucose in the body. So just be breaking it down, produce energy. If you need the energy, if you're doing some hard work or working, any part of your body needs that ATP, there's going to be excessive breakdown of it. If not, there are other pathways we're going to see, like storage and all of that. So insulin, for example, activates or increases the rate of hexokinase. AMP, which is an intermediate of ATP. ATP is the energy molecule, but before ATP, you have AMP and ADP. So if you have excessive AMP, means the body has to convert it to ATP. So it's going to encourage this pathway to use this AMP to produce ATP and so on. So the same explanation goes for all of this. So you can try to put those together and see if this makes sense for you. If it doesn't, of course, you can always ask your questions in the comment section. All right. So now we said that glycolysis is a pathway that produces pyruvate or lactate. We mentioned lactate, remember. But the lactate comes about depending on the chance of the body. So in humans, lactate is produced when there is no oxygen. Pyruvate is produced and then there's no oxygen in that particular cell. The cell is going to produce lactate. In yeast, ethanol is going to be produced. So pyruvate is converted to ethanol or lactate, depending on the cell. If it's a human cell, like red blood cells or contracting muscle, you have lactate and ethanol in yeast, for example. This brings me to a story. Not really a story, but I just want to demonstrate this. I don't know if you know of puff puff. So puff puff is a snack eaten in my country. I love it so much. I have tried to make it several times, and luckily for me, the last time I did, it came out good. But in the process of making puff puff, you mix flour, sugar, and yeast in the mixture. Of course, we know that the yeast is supposed to make the flour to rise and so on. But how does it do that? It uses the sugar that is there, glucose, it's breaking it down into pyruvate and to ethanol in the absence of oxygen, because to make it rise, you have to cover it, prevent oxygen from going in and so on. So I remember the last time I made the puff puff, I started frying and the gas got finished, so we had to wait. The one I had fried already, like few of them, was sweet. We ate it and it was okay. But the one we didn't fry, we had to leave it to the next day. It was too late to order for gas at that time, and we left it to the next day. So the next day, I fried the rest, and it wasn't sweet anymore, and it had that smell of alcohol. I remember my kid sister telling me that, why is it smelling like alcohol? She's going to get drunk from eating puff puff. And I guess you can explain that. Leaving it overnight, the yeast consumed all of the sugar to produce ethanol. That's why the next day it wasn't sweet anymore and it was smelling like alcohol. The the sugar had all been broken down into ethanol overnight by the yeast. So it's just an analogy to help you understand how this goes. But in the presence of oxygen, which occurs in human body, in most human cells that undergo aerobic glycolysis, at the end of the day, the pyruvate is converted to acetyl CoA, which goes into the Krebs cycle. We're going to see the Krebs cycle in another video. All right, so that is all for this video. If you watched, if you understood, if you liked it, you should definitely comment in the comment section. You should like the video and share with other curious molecules, of course. Comment all the answers too and all the questions that you have, other video ideas that you want us to make, other topics you want more explanations about and simplification. You should comment in the comment section below. And also, don't forget to answer the questions yourself, read other materials. If you want that, I could also refer some books and materials for you to read and watch to be able to understand these topics better. So stay blessed and thank you for watching.